I'd like to extend a warm welcome to all of you who are joining us today. My name is Damien Doherty and I'm the Editor-in-Chief of Inside Precision Medicine. And I will be hosting today's webinar entitled MinimuMM-seq Genome Sequencing of Circulating Tumor Cells for Minimally Invasive Molecular Characterization of Multiple Myeloma. This webinar is supported by Menarini Silicon Biosystems. In this proof of concept study, Ankit and his colleagues have established a novel liquid biopsy approach, MinimuMM-seq, enabling the enumeration and sequencing of CTCs to replace standard molecular cytogenetics performed on bone marrow of multiple myeloma patients. CTCs harbored the same pathognomonic DNA abnormalities as clinical bone marrow samples. Longitudinal sampling of CTCs was able to track clonal dynamics over time. These results demonstrate that genomic biomarker profiling of CTCs could be used clinically for screening and monitoring of patients with multiple myeloma in a minimally invasive manner. So let's introduce our speaker, Ankit Dutta, Postdoctoral Fellow at the Dana-Farber Cancer Institute and the Harvard Medical School and the Broad Institute. Ankit's experience lies in molecular biology, genomics, and hematology, and ostensibly in the intersection of biology and technology with the use of genomic technologies to unravel the genotypic and phenotypic basis of disease. He is currently focused on the area of multiple myeloma. And as you'll hear today, he will talk about a novel blood-based biopsy methodology and the clinical utility and indeed impact of liquid biopsies for cancer patients. Please do feel free to submit questions as we progress through the webinar. We'll endeavor to answer as many as we can in the time provided. And if we aren't able to do so, the Menarini team will be glad to make contact and answer your questions post-event. I'll now turn things over to Ankit.
So good morning. It's a pleasure to be here today to discuss circulating tumor cells and our work in the myeloma context titled MinimuMM-seq, Genome Sequencing of Circulating Tumor Cells for Minimally Invasive Molecular Characterization of Multiple Myeloma Pathology. I'd like to thank the Inside Precision Medicine team for the invitation to speak today and the audience attending here. And while it's my honor to present our research study, this has been an incredible collaboration from our labs, the Gabriel Lab, with the Getz Lab and the Silicon Biosystems team. So today I want to talk about the blood and bone marrow cancer myeloma, liquid biopsies, especially circulating tumor cells, and some of the results from our recent study, and finally finish with some of the clinical applications where we think that CTCs can really help with the diagnosis, monitoring, and risk stratification for myeloma patients. So multiple myeloma is a largely incurable hematologic malignancy which is characterized by the clonal expansion of abnormal plasma cells within the bone marrow space. And as you can see from the figure below, CD138 plasma cells crowd out the bone marrow space. Myeloma accounts for approximately 20% of deaths from hematologic cancers and 2% of deaths from all cancers. And overt myeloma is defined by clinical features defined by the CRAB criteria, and this includes hypercalcemia, renal failure, anemia, and bone lesions, due to the accumulation of these abnormal plasma cells. With the rise of genomic technologies over the last decade, there have been numerous next-generation sequencing studies performed on myeloma patient samples, showing that myeloma is characterized by extensive genetic heterogeneity. You can see in the summary plot, the CIRCOS plot on the right, that there's many somatic driver mutations, copy number abnormalities, gains and losses, and translocations, which are found across different subgroups of myeloma patients. Myeloma is an interesting cancer as it develops from well-defined precursor disease stages, which have been studied, including MGUS or monoclonal gammopathy of undetermined significance and smoldering myeloma. Patients diagnosed with these precursor conditions remain asymptomatic in their nature. So primary genetic events such as hyperdiploidy or IgH translocations are known to lead to the onset of MGUS, with further secondary genetic events such as somatic driver mutations, copy number abnormalities, and accumulation of tumor burden leading to the transformation to smoldering myeloma and overt multiple myeloma. I also wanted to highlight here that while this is mainly occurring in the bone marrow compartment, myeloma also has a peripheral blood component. And despite the development of effective therapies which have improved patient outcomes, Most myeloma patients inevitably relapse and require further lines of treatment. This highlights the need for improvement in early detection methods. So the prevalence of these precursor conditions of MGUS and smoldering myeloma have been studied and to be found in the general population and estimated to be around 3 to 5% for MGUS and 0.53% for smoldering myeloma. Screening may be especially important at the smoldering myeloma stage, which is a heterogeneous intermediate stage. You can see from the plot on the right that smoldering really captures a triphasic group of patients. One being high risk and where patients progress within the first 2 years of diagnosis, and they're being more myeloma-like. An intermediate stage where patients are more indolent. And finally, a group of patients which are more MGUS-like and don't progress for many years. A key factor here is that the diagnosis of these precursor disease stages still remains an incidental process from routine blood tests where elevated paraprotein is found, as there's no specific routine screening or monitoring tests available. And this really brings us to the clinical challenge in the myeloma setting. So bone marrow biopsy is required to fully characterize a patient's disease and is the gold standard for diagnosis and monitoring of patients. So this includes the isolation of tumor plasma cells from a biopsy sample to perform molecular cytogenetics and the FISH test, or fluorescence in situ hybridization, to detect specific chromosomal abnormalities underlying tumor biology. This can be used for the prognosis and risk stratification of patients. However, bone marrow is not always able to fully characterize a disease, and this can be due to a number of reasons, such as FISH failure, when there's insufficient tumor cells, insufficient plasma cells for the FISH test, and furthermore spatial heterogeneity, where bone marrow may be patchy, so the biopsy doesn't collect and represent all the tumor cells that are actually present. The median age of myeloma patients at diagnosis is around 69 years, and therefore patients being of more elderly age, bone marrow biopsy is an intrusive and painful process, and therefore is not feasible to be performed for repeat sampling and monitoring of patients in this way. This highlights the urgent need for robust early detection methods that are both tumor biology based and minimally invasive. Liquid biopsies are a more patient-friendly process, with the isolation of peripheral blood already present as part of the current workup of patients. One such analyte present within peripheral blood samples are circulating tumor cells, and these could represent one such alternate sampling analyte that can be used to characterize a patient's disease. This can be done in multiple ways, such as enumeration or counting of CTCs and their association with disease stage, and furthermore, the genomic profiling of these cells to detect myeloma abnormalities. So circulating tumor cells are rare tumor cells that are shed from the primary tumor site into the bloodstream. And as these are intact whole cells, they can be used for the investigation of molecular characteristics of the primary tumor. You can see on the figure on the left that this can be done through multiple omic methods, such as genomics on DNA, with whole genome amplification and the assessment of copy number abnormalities, or the detection of driver mutations, such as actionable single nucleotide variants. Furthermore, at the transcriptomic level, this could include bulk RNA sequencing, single-cell RNA sequencing, or targeted qPCR analysis for specific genes of interest. Additionally, a functional analysis, such as proteomics or in vivo modeling, can also be performed to understand tumor biology. The results of these molecular analyses can really assist at the clinical level, as CTCs can be used in cancer detection context for screening and early diagnosis. But furthermore, serial analysis of CTCs can really help with monitoring of patients and their genomic profile and how clonal evolution and progression is occurring over time. Research in the CTC space is active, is quite active, and there's many ongoing developments of technologies for the detection and capture of these rare cells present in the blood. This includes different methods that use positive selection, negative depletion or negative selection, and also size-based identification of CTCs. So circulating tumor cells in the myeloma context have been assessed with studies over the past 15 to 20 years, which have mainly focused on the enumeration of CTCs and their association with disease staging. And we've seen that with an increase in technologies over the years, an increasing count of CTC has been found across staging, with slide-based enumeration to conventional flow cytometry to next-generation flow. There's been a few studies performing molecular profiling, and these have been targeted in nature, looking at DNA for driver genetic mutations, or transcriptomic, looking at expression profiles of genes, finding that they're similar to bone marrow tumor cells. And in our study, this was one of the key aims that we had, was not only to perform enumeration, but also molecular profiling and take analysis to the next level in an unbiased way. And we'll discuss some of the myeloma studies in the next slides coming up. So studies have shown that enumeration of CTCs has prognostic value in the myeloma context, where research has demonstrated that increasing CTC numbers with disease staging from MGUS to smoldering myeloma to multiple myeloma compared to healthy donors. In the overt myeloma setting, increased CTC counts have been found with higher clinical risk group staging for myeloma, and furthermore, CTC counts have been found to correlate with clinical measures of disease burden, such as bone marrow plasma cell percentage or M-spike levels of patients. Targeted sequencing studies of CTCs have shown that CTCs match the primary bone marrow compartment, and in the overt myeloma setting, CTCs have been shown to be representative of the primary bone marrow tumor cells, with whole exome sequencing showing that CTCs harbor genetic abnormalities of myeloma, including clonal driver mutation and copy number abnormalities. And furthermore, transcriptomic profiling has shown that CTCs express bone marrow tumor markers such as BCMA, CD38, and SLAMF7. So taken together, these results suggest that quantitative disease monitoring at regular intervals may be feasible using a liquid biopsy-based test and circulating tumor cells. However, while previous studies have relied on targeted methods, there's been no studies using unbiased technologies to detect any abnormality present, and specifically this includes translocations, which were one of the key primary events of myeloma, which was discussed earlier. And this really brings us to the aim of our study, which is to determine the clinical utility of enumeration and genomic profiling of CTCs as a minimally invasive liquid biopsy-based screening method for the early detection of myeloma using whole genome sequencing. Our experimental workflow included the collection of peripheral blood from a cohort of 261 precursor and overt myeloma patients prospectively collected on the P-crowd or precursor crowd study at Dana-Farber Cancer Institute. Circulating tumor cells were enriched using the Silicon Biosystems CMMC assay kit based on CD138 and 38, with the exclusion of white cells based on CD45 and 19. The absolute count of CTCs were enumerated in each patient using the CellSearch CellTracks platform. In 51 patients, we further sorted CTCs to improve their purity before downstream whole genome sequencing and genomic profiling to detect myeloma-specific events. So circulating tumor cells were evident in the majority of precursor patients that were tested, with one or more CTC detected in 82% of all patients that were enrolled. This included 75% of MGUS patients and 86% of smoldering myeloma patients with successful enumeration. You can see on the plot on the right, we found a significant difference in the median number of CTCs between both stages, with a median count of 3 at MGUS and a median count of 23 at smoldering myeloma. A previous study using the same enrichment technology, but in the overt myeloma setting, has shown that 98% of myeloma patients are found to show CTCs in the peripheral blood. A proportion of smoldering myeloma patients collected also had bone marrow biopsy and results available to perform risk classification using the International Myeloma Working Group 2020 model. This model relies on the bone marrow plasma cell percentage, M-spike level, and free light chain ratio to assign a risk classification to patients. A higher CTC count was associated with increasing risk group based on this three-factor model, with a median count of 5, 34, and 170 CTCs detected at the low, intermediate, and high-risk smoldering myeloma groupings. We found that CTC counts were significantly elevated at the intermediate and high-risk group compared to the low-risk grouping. While enumeration of CTCs provides a correlative measure of disease burden, downstream molecular characterization can confirm tumor origin and myeloma-associated abnormalities. We first characterized CTCs by imaging and low-pass sequencing, with imaging confirming that CTCs had the correct immunophenotype with CD138 and 38P staining, and were cells of good quality and morphology. Ultra-low-pass whole genome sequencing was performed in a collection of smoldering myeloma patients that had paired CTC and bone marrow results at the same time point. Circulating tumor cells were found to harbor arm-level copy number abnormalities, which were concordant with bone marrow results from clinical testing. You can see in the example on the right a smoldering myeloma patient where arm-level copy number abnormalities are found in the circulating myeloma cells, including 1q gain and deletion of chromosome 13 and 22, which reliably match what was also present in the bone marrow plasma cells. Additionally, you can see that germline white cells show no copy number abnormalities as expected. This gave us confidence in our enrichment strategy to move forward in a wider collection of patients with standard whole genome sequencing. To determine the equivalence of CTCs with clinical bone marrow and FISH results, we performed whole genome sequencing in 24 patients where we collected matched CTCs and bone marrow samples at the same time point of clinical visit for patients. A median of 394 CTCs and 11,700 bone marrow plasma cells were isolated and sequenced. Additionally, for each patient, a germline non-tumor sample was used as a reference control. Our enrichment strategy yielded a high tumor purity result with quantification by absolute, showing a median tumor purity of 98% for CTC samples and 99% across all bone marrow plasma cell samples. We first performed a head-to-head comparison of whole genome sequencing of CTCs with clinical FISH results, showing that CTCs were able to detect 100% of clinical abnormalities that were reported. You can see in the upset plot to the left, new high-risk events were additionally found, such as 1q gain and MYC translocations, which were detected by the whole genome sequencing test, but were not found by FISH probes that were used. Overall comparison of large-scale events across all patients showed that CTCs reflect the major bone marrow clone with 100% match in key structural events that were identified. The plot on the right shows for each patient. So the upper row illustrates results derived from their CTC sample and the bottom from sequencing of their bone marrow sample. And this really highlights that CTCs are able to reliably recapitulate all resulting abnormalities that are found in the bone marrow, including primary translocation, shown on the right of the figure, and key copy number abnormalities, such as 1q gain, odd chromosomal trisomies, and deletion of chromosome 13 that were detected. Translocations that were found both by FISH and our whole genome sequencing test are shown with black box colors, whereas those found by whole genome sequencing alone are shown in green. For chromosomal abnormalities, you can see that copy number gains are shown with varying intensity in red colorway, and those where the copy number deletions are present are shown in a blue colorway. We then went on to perform head-to-head comparison of extracted FISH results with whole genome sequencing of CTC results, finding an equivalence with 100% sensitivity and specificity against all FISH probes that were tested. Compared to FISH, we found an additional diagnostic yield in 20 patients, and this is illustrated in green text in the table below. Here we can see the clinical FISH results annotated in the left column, with comparison whole genome sequencing yield in the right column for each patient. In particular, 4 smoldering myeloma patients had FISH failure due to insufficient cells for clinical testing. However, we show that whole genome sequencing is able to recover a result and reliably detect cytogenetic abnormalities that are present in these patient tumor cells. You can see below the 4 starred patients on the left, and this included key myeloma events such as primary translocations, 1q gain, trisomies, deletion of 1p, and deletion of chromosome 13. This suggests that blood-based screening of CTCs is reliable to detect alterations of bone marrow even when clinical bone marrow fails. We then went on to demonstrate the clinical utility of whole genome sequencing by collecting peripheral blood only from an additional 27 patients. Similarly, we show comprehensive detection of translocations and copy number variants of myeloma across all these patients, and we highlight here 2 key examples of smoldering myeloma patients where novel high-risk events were detected. In the first example on the left, you can see that clinical FISH testing found a primary translocation and deletion of chromosome 8, 13, and 17p. We showed that whole genome sequencing has a sensitivity to layer-in mutation, finding a mutation in the driver gene of p53. And notably, this confers a biallelic double hit event with the 17p deletion, which is a high-risk event of myeloma that would not be found by standard clinical testing, as mutations are currently not tested for. In the second example on the right, we can see that clinical testing showed the patient multiple trisomic events, 1q gain, and IGH separation with an unknown partner. We show that whole genome sequencing is able to recover this unknown result as a MYC-IGH translocation, as well as additional trisomic events on chromosome 19 and 21. And once again, the ability to find hotspot mutations in driver genes, and here we find one present in the NRAS gene. As peripheral blood continually circulates, liquid biopsies may be affected by the phenomenon of spatial and temporal heterogeneity in mutational burden. In patients with matched CTC and bone marrow samples, we investigated the clonal architecture between compartments and whether the dominant clone or preferential circulation of subclones was identified. At the genome-wide scale, mutations were found to be shared between both samples, indicating the maintenance and representation of a clonal complexity in the blood. This is exemplified in this smoldering myeloma example, where plotting 2D density plots of cancer cell fraction of mutation shows a high mutational density in the upper right diagonal, harboring primary translocation and KRAS G12 mutations found in both samples. And we also found a second high mutation cluster with a second KRAS mutation, or G13, having a vertical shift from the diagonal, suggesting that it is preferentially found in the peripheral blood sample. Phylogic NDT was used to cluster mutations into branching subclones, and indeed show that KRAS G12 was a shared clonal event, while KRAS G13 was found to be a subclonal event. And this is represented by the green and blue branches in the phylogenetic tree. This also suggests that KRAS G13 conferred additional fitness advantage for tumor cells to circulate. Finally, we wanted to address the feasibility of serial liquid biopsy sampling and whether CTCs could track disease burden in untreated smoldering myeloma patients over longitudinal time. We analyzed 7 patients at 2 time points and found across all patients stability of the major clone with clonal evolution and clonal tiding of CTC subclones over time. In this example below, you can see this is exemplified in a smoldering myeloma patient which had sampling approximately 2.5 years apart. We found a high mutational density in the upper right corresponding to the major clone at both time points, harboring 1114 translocation, and an additional high-density cluster with a vertical shift, illustrating that it is preferentially found in the second blood sampling time point, which harbored deletion 13q and an NCOR1 driver mutation. We use phylogic NDT to cluster these mutations into branching subclones, and indeed you can see that the 1114 was identified as a shared clonal event at both time points, highlighted by the green clone, and emergence of the blue clone with driver mutations of NCOR1 and del13q from 8% to approximately 80%, with extinction of the orange clone that had passenger mutations. So these data demonstrate the use of CTCs to successfully track the mutations and clonal evolution of myeloma over time in a minimally invasive manner. So the presentation so far has focused on key findings of our study and has hopefully demonstrated the clinical power of CTCs for myeloma patients. In the last section, I wanted to circle back to highlight some immediate clinical application of our novel method. So as previously discussed, bone marrow biopsy is the gold standard for diagnosis and monitoring of patients. And after a biopsy is taken, molecular cytogenetics and FISH testing can be performed to detect chromosomal abnormalities underlying tumor cells. These can be used for prognosis and risk stratification of patients. So how does the FISH test work and how can it be improved? So FISH is a site-specific high-resolution cytogenetic test for prognostically important subgroups of myeloma. So after a bone marrow sample is taken, plasma cells are enriched and isolated before slide preparation and FISH testing is performed with different probe sets. In the example below of a primary translocation of 1114, you can see when chromosomal arms don't have translocations that these light up as either red or green. However, in tumor cells where chromosomal arms do have translocation events, These probes come together and light up as yellow, as indicated by the arrows in the figure to the right. As genetic abnormalities are the most important factors present in tumor plasma cells, a myeloma panel aims to capture key prognostic events of these translocations and copy number to assess progression and survival of patients. You can see that these fall under different risk groupings, including standard risk, intermediate risk, and high risk, and include some of the events detected as shown today in our results, such as trisomies, primary translocations, gain of 1q, and deletion of 17p. A key limitation of the FISH test is that methods and panels can vary across different centers, such as not testing for all abnormalities in probe sets, or hyperdiploidy not always being assessed. A solution to this would be an unbiased genome-wide method for systematic detection of any myeloma event, such as whole genome sequencing used in MinimuMM-seq. In the genomics era, implementation of clinical sequencing to improve stratification of patients based on genomic biomarkers has a high potential. We wanted to assess whether genomic profiling of CTCs for stratification could provide a minimally invasive and repeatable solution to refine patient classification using existing models. The matrix plot below shows head-to-head risk stratification based on chromosomal abnormalities assessed by molecular cytogenetics versus whole genome sequencing. There's a lot going on, but hopefully I can walk everyone through it. So the rows are defined by important myeloma risk factors from classification models, with each column being a patient's result. Where an event was detected by both tests, the results are colored in black, and when a result was only found in CTCs using whole genome sequencing, these are shown in green. So as you can see, CTCs were able to detect all reported high-risk prognostic factors, such as primary MAF translocations, deletion 17p, or 1q gain across all patients when they were present. Risk stratification using available cytogenetic-based models showed matched patients would have been compartmentalized into the correct risk grouping when bone marrow FISH was successful, as highlighted in the bottom rows entitled Risk Group by Clinical FISH or Risk Group by MinimuMM-seq. Additionally, the recovery of FISH failures enabled the attribution of clinical risk in 3 patients, falling into high, standard, or intermediate risk classifications. These would have been missed by standard testing until the next bone marrow for patients, and this highlights the importance of robust methods for detection at diagnosis and patient visits. And as you can see, these patients harbored primary translocation, such as MAF, trisomies, and deletion 13 or 13q. So in summary, our study shows the clinical utility of enumeration and molecular analyses of CTCs and myeloma, with CTCs detected in the peripheral blood of majority of precursor patients, and a higher count in smoldering myeloma correlated with the increased clinical disease risk staging. Furthermore, molecular analyses of CTCs showed that CTCs are of tumor origin and reflect the tumor cells of the primary bone marrow. Genomic profiling using whole genome sequencing could be used to detect translocations and copy number of myeloma. Finally, longitudinal sampling of serial CTCs can be used to track the clonal dynamics of myeloma over time. And these highlight the potential clinical utility of circulating tumor cells to replace molecular cytogenetics for diagnosis and monitoring of patients in a minimally invasive manner. And finally, I'd like to acknowledge all my colleagues in the Gabriel Lab and clinical team, especially my co-authors, Jean-Baptiste Alberge and Elizabeth Lightbody, the clinical team led by Anna Cowan and Erica Horowitz, and our mentor, Dr. Irene Gabriel. Additionally, our collaborators at the Broad Institute in Dr. Gad Getz's lab, all the funding bodies that have supported our research, and finally, the patients who contributed our research samples for us to undertake this study. And thank you all for your attention today, and happy to take questions next.
So it leads me to thank Ankit for this very informative talk. We hope this presentation will provoke further ongoing discussion. And with that in mind, the Q&A is coming up shortly. So if you have a question for Ankit, please do type it into the Q&A box. Okay, folks, so we'll now transition to the Q&A. So we'll be right with you momentarily. Okay, so thank you, Ankit, for that wonderful presentation, and welcome back, everybody, to the Q&A session. We have a few questions that have come through. whilst Ankit has been speaking, which is good to see. So Ankit, we may as well dive in now and have a look at some of these questions and see if you are able to elaborate. The first one we've got that's come through says, now you mentioned enumeration in myeloma. How does this compare to solid tumors?
Thanks, Damien, and thanks everyone who's joined the call today. Yeah, so it's interesting. So obviously we're presenting on work we've done in myeloma, which is a key focus of our group, which, as I mentioned in the presentation, is a blood and bone marrow cancer. So given that we actually see circulating tumor cell numbers which are much higher than what has been seen in solid tumor context. So routinely using not only cell search, but other methods that were mentioned, such as flow cytometry or next generation flow-based methods, it's usually in the hundreds to thousands. Whereas if you look at solid tumors such as breast, colorectal, the numbers are much lower. And, that can be, 0 to 10s. And especially with cell search. So the kit that we've used was called the MinimuMM-seq kit, as was part of the methodology workflow slide. But they also have a kit, which is the CTC kit, which is based on EpCAM. And a lot of studies that have been done in those solid tumor contexts have routinely showed less than 10 CTCs. And I think that really speaks to the biology of the differences in the tumors, whereas obviously you need to have EMT transition, and have these EpCAM like these very low EpCAM cells that are circulating, whereas myeloma plasma cells, they do traffic between the bone marrow and the blood, and that's kind of what we're capturing with the high numbers that we see in outpatient cohorts that we look at.
Okay, wonderful. Thank you for that. Next one that's come in says DNA sequencing was the main omics studied. Does unbiased transcriptomics also have an important role in translational CTC work?
No, thank you. No, this is a great question, and obviously there's a lot going on in the omics space, especially not only at the DNA level, which was our interest, but transcriptomics and even spatial and other things that are going on in the field. So we've really focused on DNA sequencing because our immediate kind of goal was to look at kind of how can we have a piece of work that has immediate translational impact, especially for patients. So as was mentioned in one of the concluding slides, risk stratification is really based on underlying chromosomal or DNA abnormalities of these plasma cells, tumor plasma cells. But definitely transcriptomics, whether that's bulk RNA or even single cell RNA sequencing, looking at these CTCs definitely has an important role, which gives us something different to what we looked at in our study. And that would really be surround the kind of the biology and the mechanisms of these circulating tumor cells and what's really allowing them to gain a phenotype that lets them come out of the bone marrow and circulate. And I think one of the key things here is, and as you could see in the cohort of patients that we analyze, we had a lot of precursor patients, so MGUS and smoldering, which are very early stages compared to overt myeloma, which is the end stage of disease, or the overt symptomatic stage of disease. So whether, we could derive transcriptomic based signatures, which kind of give the biological underlying a more aggressive disease, and whether we could apply these kind of signatures and map them in the early stages to kind of identify patients that may have maybe more progressing rapidly or more having these aggressive disease phenotype, to get them early, and then obviously have closer monitoring or intervention, which is feasible to give that at the earlier stage.
Okay, brilliant, wonderful, thank you. Another one that's come in here says the study cohort investigated precursor multiple myeloma patients. Thoughts on the significance of finding CTCs present at these early stages of disease?
This is a great question, and I think that was one of the other things where we tried to extend on the application of this MinimuMM-seq kit. So it was obviously been under development for a few years, and the first major cohort which was looked at was overt myeloma patients, and that was in collaboration with the Multiple Myeloma Research Foundation. So a very large collection, like over 500 myeloma patients that were enumerated. And obviously, given that it's overt stage of disease, we expect that, there's some clones, plasma cell clones, that are obviously gaining a more advanced and aggressive phenotype, and then, ability to circulate. So especially at the precursor stages disease, it's, I guess the thought usually around CTCs is they're, obviously associated with dissemination and metastatic potential. But being a precursor stage of disease, we don't really anticipate that because we don't have this burden of disease, which is that great at that stage yet. So I guess our thoughts around the significance of finding CTCs this early, I guess are two things. So one being that some of these precursor patients that we've been collecting, obviously these are new patients that have only been monitored clinically by clinical measures in the blood, that it could be that these are patients that are starting to progress and they're having these disseminated CTCs in their blood. Imaging isn't part of the routine of these early disease stages, so maybe they have secondary site seeding that we haven't seen yet because we don't have that workup or any imaging to look at. So that's one point. And the other thought could be that, and this has been shown by next generation flow in cohorts that those groups have analyzed, is that CTCs seem to follow this kind of timing pattern, and with release, specific release from the niche with specific HSCs, and especially in the early phase of time during the day. So I think it was 4 a.m. to 9 a.m. where they saw the highest numbers of CTCs, but also kind of when looking at markers by flow-based methods. So yeah, one could be just due to the circadian rhythm of how things are going on with CTCs and HSCs, but the other could be that these patients are patients that are progressing and going on towards having transformation. But consider we had single time points or a very few patients that have multiple time points. We're not able to address this, but that's definitely an important point that going forward, we'd love to think more about and address.
Okay, thanks, Ankit. Got another one here. Spatial heterogeneity is a feature of myeloma. How do you know if CTCs are from the primary site or secondary site seeding has occurred?
Now, this is another great question, really important for the myeloma context. So at least for the cohort that we did, especially the 24 patients where we showed that we actually collected bone marrow sample at the same time point as collecting their blood sample to enrich for CTCs. When we perform whole genome analysis, so we use one tool which is able to map the VDJ reconstruction. So basically we can use immune repertoire profiling to look at the underlying VDJ of the clonotype and able to match them for the CTC sample and the bone marrow sample for each of the patients. And when we did this across those matched patients, we can see that there was 100% match. And that gives us the indication that the CTCs that we were looking at come from the primary tumor of the bone marrow of that patient specifically.
Okay, wonderful, thank you. Another one coming in here, they're flying in thick and fast. Other than myeloma, in which different cancers is this technique applicable clinically?
No, so this is a good question. So as we mentioned in the methodology, so we've specifically used a kit that's developed by Silicon Biosystems. So it's actually developed specifically for circulating multiple myeloma cells. So this would mainly be for myeloma and its precursors, so smoldering and MGUS. So the kit is really devised on immunophenotype of plasma cells, which are the kind of tumor cells that we look at. So CD1, 38 and 38. And then we have 19 and 45 as well. So this kit specifically would be for myeloma only context, but the CTC kit is based on EpCAM, so it could be used in a wide range of solid tumor contexts as well.
Okay, wonderful. So for genome sequencing, we need minimum of 2 UG of gDNA. So how much total CTC required for whole genome sequencing?
No, this is a great technical question, and this is something that we in the first years of kind of work up the methods of what we were doing was really a key thing we were trying to address. So I think, yeah, in previous years, and we have these kind of standard whole genome sequencing kits, Illumina, etc., have required quite high yields of input of template DNA. But in our method, we actually used a enzymatic-based protocol, which came out maybe 2 years ago. And there's a really good paper from Professor Peter Campbell's group, but it's a low input template method where you can do library construction. So from here, the lowest limits we had were about 300 to 400 cells. So from that, we were able to get adapter ligated libraries that we're able to amplify enough for PCR to get a final library that was sequenceable. So that in the range of, as low as 2.5 to 3 nanogram, we were able to work with.
Okay, brilliant, thank you. Another one here, what type of tube is used for CTC enumeration? What is the turnaround time for whole genome sequencing from enumeration to sequencing?
This is a great technical question, especially for the clinical applicability. And I think a lot of the power of the work that we were able to do comes from a lot of the R&D development that's been done by the Silicon Biosystem team. But as part of the CMMC workflow, there's actually a specific preservative tube that's used. So it's called Cell Rescue, but it stabilizes the blood collection and CTCs, and you can run them up to 4 days later. Obviously, you want to run them as soon as you've collected, but it does give that time to say if you're getting patient blood from external sites, for that to be shipped in, and you can run that on the machine a day or two later to perform the enumeration. So in the second question, in terms of turnaround time for doing whole genome, so obviously we work with not only our lab, but also our collaborating labs over in the Broad Institute. So not only Professor Getz's lab, but also the Broad Genomics Core. So going from enumeration to sequencing for a batch of about 8 patients, so consider that a NovaSeq, full NovaSeq run, that would take us about 2 weeks. 2 to 2.5 weeks, and obviously that's dependent on the genomic platform workflow. But given if this was like a full in-house protocol that was done end to end, I think within 2 weeks we could go from patient collection to having a final output of result and an interpretation.
Okay, wonderful. Thanks, Ankit. What about sensitivity in multiple myeloma? For example, can CTCs be useful for detecting minimal residual disease?
No, great question. So I think that's a key point, and one of the limitations of our study is that at this point we focus mainly on untreated diagnosis stages of MGUS, smoldering, and some myelomas. But obviously, one of the other key things, not only for detection of disease, is also monitoring, and obviously monitoring response to treatment. So I think that's one of the next kind of aims or thoughts that we had, obviously, that there's application for potentially identifying minimal residual disease and the duration of MRD by CTC. It's not something we've addressed using the CMMC kit, but there's been work done by next generation flow-based methods. And I think one of the key papers, at least when they used that technique and showed in that study, is that there's still a kind of high false negativity rate. So when MRD was present in the bone marrow of these patients, when they collected blood for CTC analysis, they did not find any CTCs present. So I think there's a lot more work to be done in that space, but definitely something critical for myeloma, and obviously the array of treatments that are available, and to look at not only duration, and then whether we can kind of also have a minimally invasive test for these patients that are in these kind of critical stages. Okay,
brilliant. Thank you. What differences are seen in CTC profiles in IgGMA and IgA myeloma?
No, this is a great question, really surrounding the biology. So for the majority of our patients, because we focus on those patients that are MGUS, but those that have the trajectory towards a myeloma phenotype, we don't usually collect IgM patients, which go on to Waldenstrom. Specifically off the top of my head, I cannot remember the distribution differences between IgG and IgA patients that we've collected, but I can definitely circle back and see, but not off the top of my head, I don't remember any kind of major or substantial differences between partitioning groups based on their immunoglobulin.
Okay, all right, good man. Thanks, Ankit. Another one here. What is the depth of MinimuMM-seq, or how many Gs do you seek sequencing 1-MMCTC. Sorry if I've mistranslated that, but
the first part I can answer. I'm not sure how many Gs do you sequence in one myeloma CTC. I'm not sure that part. But I'm assuming the first part of the question is asking about the depth of sequencing, maybe.
Yeah.
So across the cohort that we had, so the 24 paired patients, but also the 27 patients that had blood only, our median coverage, I think, was around 20 to 25x. And due to the purity that we had of the sample, due to the way we were enriching and sorting, this was obviously very sufficient to look at mutation and structural variant detection.
Okay, brilliant, thank you. And then we've got one more here that says, what is the success rate for whole genome sequencing? Were you able to successfully sequence every single sample you intended to?
No, this is a great technical question. So for the collection of patients that we had, we were able to successfully sequence each patient that we intended to. And I think this goes part of the build-up of the workflow. So obviously we mentioned that we were first performing this enrichment on the cell search platform. So that obviously lets us have the enumeration of each of the patients that we've collected in the cohort. So that actually we can see what the absolute numbers of CTCs are present. So based on those, we were able to choose which patients we knew had higher burden, which met the kind of metrics that we'd worked up in our pilot stages of how much DNA is required to actually perform this whole genome sequencing library and sequence, as part of one of the earlier questions that was asked. Obviously, patients that fell below that, we couldn't perform whole genome, and obviously we have an enumeration count on them. But I think this also goes to the fact of having this kind of, and I mentioned in some of the intro slides, is that a lot of study had focused on just enumeration and some targeted molecular profiling, but having both enumeration and genomic sequencing, you're able to follow these early stage patients just with enumeration. Obviously, they may have low CTC numbers, which is obviously great for the tumor biology and the patient itself. But as these numbers start to increase, you can kind of observe these in real time with kind of repeated blood sampling and CTC enumeration. And once it kind of crosses a threshold of the yield required to actually do genomic profiling, then the patient sample can quickly be moved on to that to return a result. So I think it's kind of like a partition workflow, which kind of is very streamlined for early stage patients, but also patients that are progressing or late stage patients. It worked really well in that way. And I definitely see that if this was going into a clinically validated method that was used, then I would say that that would be a really good way to work it, as you'd be able to get both results on both sides and obviously make those decisions to proceed with the profiling, or whether you'd have to wait until the next visit and biopsy or blood biopsy required.
Okay, wonderful, thank you. Very well comprehensively answered all of those, so you've done very well there. Quite a few that came in, testing different parts of your knowledge there, so that's fantastic. Well, listen, that's literally all the questions that have come through. So if you do have any further questions, please also feel free to submit those, and you can obviously reach out to Ankit or, of course, Menarini. I'd just like to thank Ankit for delivering a fantastic, stimulating, thought-provoking talk, and of course for Menarini for supporting today, and of course to all of you out there that have attended today. So we're very, very grateful for you to be with us today. So this will be available on demand in the next sort of 24 hours, and it will sit on the website for a good 6 months. So please do, you know, if you need any more information, as I said, please do reach out to either Menarini or to Ankit. But it leaves me to say thank you very much for today's webinar. We've enjoyed having you, and we look forward to having you again in the near future for another installment of an Inside Precision Medicine webinar. So with that, I wish you all a very good day, and goodbye.